let us complete the concept of meiosis by this session. Meiosis is a reduction division. It consists of meiotic division 1 and meiotic division 2. Meiotic division 1 already finished. This is called reduction division, right? Reduction division. It consists of prophase, that is a prophase 1, then metaphase 1, anaphase 1 and telophase 1. In the prophase 1, there was leptotin, zygotin, pakitin, diplotin and diakinesis. After that, it will enter here, later follow. So the result, 2 haploid cells. State is N. Whereas a cell which is entering the meiosis 1 should be diploid. So that this is not the germ cell, that is meiosis is incompleted, I can say. When these haploid cells, result of the meiosis 1, are entering the meiosis 2. Meiotic 2 is homotypic division. Understand? Whereas meiotic 1 is heterotypic division. Simply, entire the meiosis is actually heterotypic division. Heterotypic division because of the meiosis 1, because reduction taken place here. Here no reduction. Chromosome number never becomes half. Here chromosome number becomes half, but here not. If you understand the differences between meiosis 1 and meiosis 2, it will be very easy to understand further concepts. So homotypic division, why I said homotypic division? Here N type of cell is entering and providing N and N. Whereas in the mitotic division, 2N type of cell is entering and 2N type of cells are coming out. But there is a 2N, but here is N. But both are same type. Same type of cell is entering, same type of cells are coming out in case of mitosis and meiotic 2 division. That is why here homotypic division. Once you call back your knowledge from the meiotic 1, here 2N is entering but N type of 2 cells are coming out, so that the cell which is entering type will be changed in the yield is called hetero. I said hetero is different. Homo means same. So haploid cell enter and 2 haploid cells will come. But here the matter is how many haploid cells are entering? 2. So one N gives 2 N's, another N also provide 2 N's. So both the cells, both the haploid cells undergo the meiotic division, result 4 cells. These all 4 are called gametes. Gametes are always in haploid state. Very, very important thing. Whether the plants or animals, gametes are always haploid. How many gametes will come through the meiotic division? 4. There is a difference between mitotic and meiotic. From the mitotic division, 1 cell gone. Completion of the mitosis, how many cells are coming? Only 2. Once a cell, diploid cell, enters the meiotic division, completion of a meiosis. Completion of meiosis means 2 steps, meiosis 1 and meiosis 2. Then answer is 4 cells. When reduction taken place, incompletion of the meiosis, result is 2 cells. But in the question, completion of meiosis is there, then answer is 4. All the 4 are haploid cells. So what are the steps involved in the meiotic 2? Prophase 2, metaphase 2, anaphase 2 and telophase 2. Prophase 2, metaphase 2, anaphase 2 and telophase 2 of the meiosis 2 similar to the mitotic division. So prophase 2 of the meiotic 2 division is similar to prophase of mitotic division. what has happened there? Disappearance of the nuclear membrane, and chromosomes are scatteredly arranged, and centromeres are gone to their poles. Then next, in the metaphase what happened? Metaphase plate has formed. All the chromosomes are comes at the center of the cell. In the anaphase 2, what happens? Just like the anaphase of mitotic division, equal division, no? So they are pulled towards their poles. But here chromatids not remain same. Okay? Then telophase, nucleic membrane again formed, centrioles are disappeared, and chromosomes become chromatin. Understand? Recondensation over there. Here the decondensation, here the recondensation, here the decondensation. So chromatin becomes chromosomes over there, and chromosome become chromatin here. Then two complete cells were formed. Nuclear division taken place, that is called karyokinesis. Finally, cell wall depression takes place. In the plant cell that is different, in the animal cell that is different. You have seen just like the mitotic division. Okay, we can say meiotic 2 is equal to mitotic division. No even small change. So result 4 cells. So one cell go through the mitotic division, result 2 cells, which is very faster. One cell gone through the meiotic division, result 4 cells, which is very steady process, slow process, because of lots of steps. Sometimes it takes place by years also. In further classes we will discuss. So from this session, you have to understand the concept of both mitotic and meiotic. Both meiotic 1 and meiotic 2. From those two questions, 10 questions you have to answer in the assignment. For better understanding, I am leaving a visual over here, followed by the good assignment. Thank you. All the best. In the next class, we are discussing the differences between mitotic and meiotic completely.
All organisms add new individuals to their population by the process of reproduction. And this can be done by either of the two methods, that is, asexual or sexual mode. Asexual reproduction is the preferred style of lower organisms and is performed by mitosis. And how do sexually reproducing organisms increase their population? That's the right answer. Sexual reproduction is aided by meiosis. We've seen the process of meiosis in detail in the previous videos. Let's summarize the entire process in this video. Let's begin with the preparatory phase, that is, the interphase first. This cell goes through 3 phases, namely G1 phase, S phase and G2 phase in order to grow and prepare itself for the actual division. And can you tell me what happens to the genetic material during the interphase? That's correct. The DNA is replicated to make an identical copy of each chromosome. On similar lines, other organelles of the cell also get duplicated in this complete phase. Among all the organelles, the duplication of one organelle is equally important as the replication of DNA. And which organelle are we talking about? Yes, it's the centrosome. And why is the duplication of centrosome so important? Because this organelle contains centrioles which form the spindle fibers needed for separating sister chromatids later. Now, this perfectly grown cell is all set to divide and entered the meiotic phase. According to the order, the cell will undergo karyokinesis and that will be followed by cytokinesis. Let's dive deeper into this world of meiosis. Meiosis is carried out in 2 rounds called meiosis 1 and meiosis 2. Why is that so? That's correct. To obtain 4 haploid cells with half the number of chromosomes compared to the parent cell. Let's begin with meiosis 1 first. Meiosis 1 is divided into 4 stages. They are prophase 1, metaphase 1, anaphase 1 and telophase 1. We will list down the changes observed in the cell during these stages step by step. Let's begin with prophase 1. It's the longest phase of meiosis 1. The phase is further divided into 5 substages. They are leptotene, zygotene, pacotene, diplotene and diakinesis. Leptotene is the first substage. In this phase, the cell begins condensation of the chromatin, making it short and compact. The next stage after leptotene is the zygotene substage. Here, the homologous chromosomes form a pair called the synapsis. The new structure that is formed after the union of two homologous chromosomes is called a tetrad or a bivalent. After forming bivalents, the cell enters the next substage called the packetene. Here, the bivalents exchange gene segments between the non-sister chromatids of homologous chromosomes. And can you tell me what this process is called? That's the right answer. It's called crossing over. And the location where the exchange takes place is called chiasma. After successfully interchanging the genes with one another, the cell enters the next substage called the diplotene. In diplotene, the bivalents unbutton themselves, resulting in the separation of the two homologous chromosomes. But here, the tricky part is that the homologous chromosomes are still anchored at the chiasma. Finally, in the last substage called the diakinesis, the homologs get completely unhooked and regain their individual identity. The nuclear membrane and the nucleolus completely disappear by the end of prophase 1. Now let's move ahead with the next stage of meiosis 1, that is metaphase 1. The homolog pairs align on the equator of the cell with random orientation. This arrangement is called the metaphase plate and is a result of independent assortment. In the successive phase, that is the anaphase 1, the homolog pairs separate from each other and move towards the opposite poles. Finally, the cell undergoes the last phase, that is the telophase 1, along with the first cytokinesis. The two new cells obtained are now haploid in nature. The telophase 1, followed by cytokinesis, helps in the formation of a nuclear membrane surrounding the chromosomes and a nucleolus as well. The daughter cells are haploid, which are formed from the diploid parent cells. Chromosomes, however, are still seen attached to the sister chromatids. So, in order to separate the sister chromatids from each other, each haploid cell undergoes another round of division. But before moving ahead, the haploid cells enter a small resting phase called the interkinesis. It's a short phase with no activities involved. After going through this phase, the cell is all set to undergo the second round of meiotic division. The next round is called the meiosis 2. It also includes 4 stages. In other words, meiosis 2 can be considered as the mitosis of haploid cells. The first phase is the prophase 2. Here, the nuclear membrane and the nucleolus begin disappearing. The chromatid condenses to become short and compact and gains a typical chromosome structure. In metaphase 2, the chromosomes align at the equator and the spindle fibers attach to the respective sister chromatids. In anaphase 2, the chromatids separate from each other and move to opposite poles due to the contraction of spindle fibers. Finally, in telophase 2, the chromosomes decondense. The nuclear membrane envelopes the chromosomes and the nucleolus also begins to reappear. The chromatids form the daughter chromosomes at both the poles. Last but not the least, cytokinesis helps divide the cell into two. Since 2 cells have simultaneously undergone meiosis 2, we get 4 haploid cells at the end. The haploid cells are also called gametes and are genetically different from each other. And why is that so? Genetic difference mainly occurs due to recombination between the chromosomes, which takes place during meiosis 1. And with this, we come to an end with the concept of meiosis.